Hello students, today we'll start up with the class. In the last lecture we have discussed regarding the cell introduction and cell discovery. Along with that I have told you that the cell topic will be discussed in three major subheadings. One is the types of cells, and the second one is the structure of the cells, and the third one is the function of the cells. So all these three subheadings will be discussed in detail. First we will take up with the type of cell. So in type of cells, mainly we have two major type of cells. One is the prokaryotic cell and the other is the eukaryotic cell. So these are the two major basic type of cells that we see in the biology. So first we will discuss with the prokaryotic cell. What is a prokaryotic cell? Here pro means primitive, and karyon means nucleus. So this is a primitive nucleus. It is said to be primitive nucleus. Why it is primitive nucleus, that we will see. If you observe, this is a cell and this is a cytoplasm. And we all know that the cell contains the major genetic information stored in the form of DNA. So this is, let us say this is the DNA. So how is the DNA here? If you observe, the DNA is directly suspended inside the cell cytoplasm. There is no boundary, or there is no compartmentation of the cell here. Directly the genetic material DNA is present inside the cell. And such type of cells are known as prokaryotic cell. Yes, definitely there is no nuclear part or nucleus, but still the content of the nucleus is present, right? So DNA, it is actually present inside a nucleus for the eukaryotic cell. So compared to the highly evolved eukaryotic cells, here the DNA, genetic material, it is present suspended in cytoplasm, and hence we say it is a primitive nucleus. Okay, though the nucleus is absent because of the presence of DNA, we call it as a primitive nucleus here. And coming to the eukaryotic cell, here U means true, karyon means nucleus, right children? U means true and the karyon means nucleus. So if you take the diagram, a small representation to make you understand the difference. Here again the cell cytoplasm is present. Let us say this is cytoplasm. And now if you observe, there is a compartment. There is a small compartment, or there is a small chamber inside which you have the DNA. Okay, so this is a separate entity or a separate chamber present inside the cell cytoplasm. Okay, so this is known as nucleus. Nucleus contains DNA, and this outer layer is known as nuclear membrane. So this is nucleus containing the genetic material DNA, and this outer layer, the boundary, it is known as nuclear membrane. And if you see the difference, here the DNA is directly present and here the DNA is present inside the nucleus. So this is the major difference which made the cells into two different categories. One is the prokaryotic cells, primitive nucleus-like cells, and the eukaryotic cells, which are advanced or evolved organisms, where we have a separate nucleus to store this genetic material, that is DNA. And coming to the most important terms, we have two important terms which we commonly use in biology, children, that I want to tell you. We use two terms, that is cytoplasm and sometimes we call protoplasm. So what is the major difference here? Cytoplasm means the cell content. For prokaryotic cell, we always call it as cytoplasm. But for eukaryotic cell, we can say cytoplasm or protoplasm. What is the difference? Here, the cytoplasm means the fluid part that is present outside the nucleus. It is known as cytoplasm. So this part is known as cytoplasm. Then what is protoplasm? Protoplasm means the nuclear fluid content that is present inside the nucleus along with the fluid part that is present inside the cell. Both together constitute the protoplasm. So what is protoplasm, children? Nucleus and nuclear material as well as the cellular material, they together constitute the protoplasm. Okay, so cytoplasm and protoplasm, both nucleus along with the cytoplasm, we call it as protoplasm. There is no question of protoplasm here. Why? Because nucleus is totally absent. So we simply say cytoplasm in the case of the prokaryotic cell. Now quickly we will see the examples of prokaryotic cells. As these are the most primitive cell types, the primitive organisms are present here, or you can say the simple organisms are present here. What are the most simple organisms present on this earth surface, children? That is the bacteria. Along with the bacteria, we have mycoplasma. So the bacteria and the mycoplasma, they are considered as the primitive cells, that is the prokaryotic cells. They have this primitive kind of nucleus. And this bacteria and mycoplasma, they are exclusively unicellular in nature. They are unicellular in nature. Unicellular means they have only the single cell. It is a structure and function of the organism. Cell means, for unicellular organism, cell means organism. Cell means organism. Yes, the cells are dividing. Many cells are produced from a single cell by division, but still, if you observe, each cell indicate an organism. Means one organism, one cell. One cell means one organism. Though the division is taking place, the cell is producing or it is forming an organism. So complete form and function. Single cell means it is the complete form and function of the organism. Okay, and if you see for eukaryotic cells, we have many organisms which are listed in this eukaryotic cells. One, we have protista. Two, we have fungi. We'll discuss these organisms in detail in later chapters. And next we have plants. And we have animals. Humans are included here. Yes. So all these organisms are included under eukaryotic cells. What I have told you, the unique feature of prokaryotic cells, they are exclusively unicellular in nature. No doubt if prokaryotic cell is being discussed, you have to keep it in mind that the organisms are unicellular. But if you observe for eukaryotic cells, we have both the uni as well as multicellular. So which group of organisms are unicellular eukaryotic cells? Can you tell me, children? That is protista. So this is unicellular eukaryotic organisms. And coming to all the three other groups, the majority of the organisms in eukaryotes, they are multicellular. So these are multicellular in nature. So here the multicellular organisms, they are placing a major role, and they are highly evolved organisms compared to the protistants. Eukaryotic cells, they have both multicellular as well as unicellular organisms, and the prokaryotic cells, we have only unicellular organisms. Now, coming to the multicellular organisms, we are not discussing with the unicellular organisms in prokaryotes or eukaryotes, because what I have told you, the complete life of the unicellular organism, it is spent as a single cell. Any activity, any function, it is completely done inside the single cell. So there is no complexity here. But for multicellular organisms, we have little complex structures, or you can say there is a little transformation and modification of the body from cells to different levels. That we will discuss in the next part. Yes. Yes. Now we will see how this multicellular organisms have been modified to form different structures. Let us see with few examples. So first, any organism, it may be a unicellular or a multicellular, the life will always begin with a single cell, right? So cell is the basic unit and the life begins with a single cell. See, for example, if we take the case of human body, the human body, the organism, it is developed from a single cell which is known as zygote. How the zygote is formed? Sperm cell from father and egg cell from mother, they come together, they fuse together to form a zygote in mother's womb, right? So here the zygote is a single cell structure. Then what will happen with this zygote, children? This zygote will undergo multiplications, divisions to form a complex organism, that is our human body, right? So how this organization is taking place at different levels, that we will name, okay? So now the cell, let us say the zygote, okay? The cell, then what will happen to the cell in the next level? The cell will form tissues, okay? Tissue level, and the tissue is again converted or transformed into tissue system. And tissue system is again forming organs. Organs are forming organ systems. Okay? So here, this is the level of complexity. Based upon the complexity of the organisms, the multicellular organisms, the body is transformed into different, different systems here. So if you see this with an example, if we compare this with an example, it will be easier for you. So first we will take the case of the plant cell or plant cell. How the cells are converted to tissues? Many cells, group of cells, they come together, they bind together to form a mass which is known as tissue. Group of cells will form tissue. Then how do you define a tissue? Group of similar cells having similar structure and function is known as a tissue. So let us say, in the case of plants, the different tissues that we see is parenchyma, collenchyma. We have different tissues, children. Parenchyma mainly for storage, collenchyma for support, and again it also help in the storage of food material. And we have elasticity And next we have sclerenchyma. This will give rigidity to the plant body. Okay, so hard structures of plant body, it is mainly made up of sclerenchyma. So all these are different type of tissues present inside the plant body. So what will happen to tissues? All these tissues, they will form tissue system. They may be present at different places. They are distant from each other, but still they are coordinated to form a system which is known as tissue system. They are different, the tissues are now different, but still they are coordinated to do certain coordinated functions. That is known as tissue system. So let us write the tissue systems that are present in the plant. We have only 3 tissue systems in plant body. One is the first one is epidermal tissue system, ETS. Shall I write it? The full form. We don't have place here. Let me adjust. So I am writing it here, children. Epidermal tissue system. We have ground tissue system, and the third one we have vascular tissue system. Okay, tissue system. So these are the main 3 major tissue systems that are present in the plant body. Epidermal tissue system, it is the external layer, just like our skin, it is the external layer for the plant body. It is mainly helping in the protection. It gives the protection to the plant body. Next we have ground tissue system. It will be helping in the storage, preparation of food material and storage, support. And next we have vascular tissue system. Vascular means it is like circulatory system in human body. Here different minerals and you can say ions, hormones as well as the water, everything it is transported through the vascular tissue system. So these are the 3 major tissue systems present in the plant body. Now what happens? All these tissue system, 1 or 2 combination or all the 3 systems, they are present inside a particular structure which is having a specific function, that is organ. Okay, so what are the organs present in the plant body, children? We have leaf, we have root, we have stem. Okay, so all these are organs, main organs of the plant body. So all these are combination, 1 or 2 or you can say all the 3 can be present in the each organ and they have a specific function. So if you observe the modification or the transformation, it is being increased, or you can say the differentiation is being increased from each level. After this, all these organs again they are coordinated and they are functioning to form an organ system. We have only 2 systems in plant body. One is the shoot system and the other is the root system. Shoot system is nothing but the aerial part of the plant body which is present above the ground, and the root system is which is present below the soil. Okay, so these are the 2 major organ systems present in the plant body. In the same way, if we take example of animals, we will see one of the example. Cells, again we will say muscle cells. We have different type of cells in human body. So highly evolved organism on the earth surface is human body. They have complexity. Based upon the complexity, the organization is also different compared to the lower organisms. So muscle cells, let us say muscle cells, they are having certain tissues. Muscle cells gather, they come together to form tissues like smooth muscle, smooth muscle tissue. We have other tissue, cardiac muscle, cardiac muscle, and we have smooth cardiac and skeletal, we will say skeletal muscle. So all this will come under the muscular tissue system. That is, they are mainly helping in muscles. How they will help the human body? They mainly control and coordinate the locomotion, or what you can say movement of the body. By contraction and relaxation, they mainly control the movement of the body. Okay. So these are the different type of tissues, muscle tissue, smooth, skeletal or cardiac muscles. Few examples I have taken. We have different type of tissues in human body, and now out of these two we are going to discuss regarding the excretory system. So I am telling you which muscle tissues are gathered to form the tissue system. Here smooth muscle as well as skeletal muscle, they are in turn forming, they gather together to form the tissue system. Smooth and skeletal muscle, skeletal tissue system, they will be forming bladder organs. Bladder organs. What are bladder organs? Kidneys. We have urethra. Okay, so all these are the bladder organs, and all of these organs, they are again coordinated to form the excretory system. Organs are different, their place is different, but still they are coordinating for which function, children? That is excretion. They form excretory system. Okay, is it clear? So if you observe both the examples I have given here, and now I will tell you the organisms which are having the multicellular nature, to which level they will be showing this modification or differentiation, that we will discuss. The first organism I have told you, that is protista, but they are unicellular. In the case of eukaryotes, we are not discussing with that. We have three organisms which are coming under the multicellular category: plants, animals, as well as the fungi. So in fungi, we have loose tissue organization. So we don't have any tissue systems or organs and organ system. Fungi will be having the tissue-like organization, not a proper tissue also, it is a loose tissue. Okay, fungi. And for plants and animals, we have the advanced organ systems are also present. But if you see the organ systems of plants, we have only few. Why? Because compared to animals, they are again lower organisms. And here, highly evolved animals as well as human beings, we have numerous systems. For example, in humans we have nervous system, we have circulatory system, excretory system, right? Lymphatic system, controlling immune system, circulatory, that is blood, everything, and we have nervous system, brain, regarding brain, that is all the different type of systems are present. They have different organs coordinating, hormones and muscles, tissues, all are different, but they are coordinated and they are helping the organism in different ways. Okay, so this is regarding your multicellular organisms and their organization. So these are the different levels of organization. Okay, is it clear, children? Next, we will move on to the next topic. So we have discussed with the differences of prokaryotic and eukaryotic cells. Next, coming to the detailed structure of a prokaryotic cell. Let's begin with the prokaryotic cell in detail. So how the cell structure has been studied, that we have discussed in the introduction part. I have told you the microscope is an excellent tool for the cell biology, and in microscopy we have light microscopes, compound microscopes, but in order to study at the molecular level or at the cellular level, we need a special microscope that is known as electron microscope, right? So we have electron microscope, children, which is most important to study the cellular structures. Okay, cell organelle is cell as well as the detailing of the cell. An internal organelle is still nucleus we can observe with the help of the electron microscope. So now we will see the prokaryotic cell. We'll do one by one. So what I will do now, I will be drawing the structure of a prokaryotic cell that you will draw in a page, okay, and you label it in one page, and each of the part, the detailing of each and every component you will do in the other page. So the detailing which I will write here, that you will do in the other page. Only labeling and the diagram will be in one page. Okay, clear? Now let us see. How is the prokaryotic cell, children? Prokaryotic cell is nothing but the bacteria. We are drawing here. We'll try to make a nice one. Right. Okay, not bad. Okay. So if you see the prokaryotic cell, I am starting with the outermost layer. What is the outermost layer of the bacterial cell? So all these terms are new. So you have to, by heart, you have to learn. You have to repetitively, you have to learn, then only you can memorize it. Okay. So the first and outermost layer of bacterial cell is known as Glycocalyx. Glycocalyx. Okay. What it is, children? Glycocalyx. Not all bacteria. Few bacteria have this outer cover, outer protective layer, that is known as Glycocalyx. Then let us see the chemical composition and how it looks like. Okay, Glycocalyx, it is mainly present in pathogenic bacteria. It is the characteristic feature for certain pathogenic bacteria. Pathogenic bacteria means the bacteria which causes disease or infection in the host body, right? And this Glycocalyx, now if you see the structure of Glycocalyx, it occurs in two ways. If the Glycocalyx is loose and thin, we call this Glycocalyx, or the Glycocalyx is renamed as slime layer. It is known as slime layer. Alright. If the Glycocalyx is tough, hard and rigid, if it is tough, everything is same, you can say tough and hard, it is known as capsule. This is highly impermeable, children. It is very tough and hard. Rarely it will allow certain substances to pass. It is almost impermeable in nature. Impermeable means it will not allow anything to come inside the cell. So these are the two versions of the Glycocalyx. If it is loose and thin, we will call it as slime layer. Tough, hard, rigid, we call it as capsule. And chemical composition. Coming to chemical composition. Chemical composition of Glycocalyx. The major substance that is present in the Glycocalyx is sugars. Those are sugars. Biological term, we call it as saccharin. Saccharin means sugar. And again, these are classified into different categories. If one sugar is present, we call it as monosaccharide, 2 disaccharide, tri. In that way, if more than 10 sugar units, they combine together to form a compound structure, we call it as polysaccharide. Polysaccharide. Polysaccharide means more than 10 units of sugar. Okay. So here the major chemical or the major component of the glycocalyx is the sugars, saccharin, and those are complex sugars. They are not simple sugars. Simple means 1 or 2 units are present. Here we have complex sugars made up of more than 10 units. We call them as polysaccharides. Not only the sugars, they have combination with other chemical molecules. Okay. If you see, they are made up of glycolipids, phospholipids, they even have glycopeptides. Glyco, in biology again, you have to remember children, glyco means sugar. Saccharin is also sugar, glyco is also sugar. Okay. Glycolipids means they are made up of sugars plus fats. Lipids means fats. Phospholipids, here phosphate is there. Phosphate along with fats are present. For some bacteria, the glycocalyx is made up of phosphate and fat, and some they are made up of sugar and fat, and some they are made up of sugar and protein. Sugar and protein. Okay. So this is regarding the chemical composition. Even if you observe for both consistency, it is different, and for chemical composition, it is different. So highly variable structure. Glycocalyx is highly variable structure. It is not conserved. It is not constant. It is highly variable. From bacterium to bacterium, it will be changing. Mainly, the chemical composition, the change in chemical composition is mainly dependent upon the external environment, or you can say in the medium in which it is growing. Okay. So, based upon the nutrient availability of nutrients, based upon the availability of the nutrients, the bacteria will be producing the glycocalyx based upon the availability of different substances. So whatever substance that is available in the medium or in the external environment, that it will pick, and with that it will make the glycocalyx. Okay, so this is highly variable. It is not conserved or constant. And the thickness, even if you observe, it is different from bacteria to bacteria. The thickness, the consistency, or the chemical composition is highly variable for glycocalyx. So it is highly variable. Okay. Then glycocalyx is present, that is okay. But what is the significance of glycocalyx? Why it is present for bacteria, that we will see. I will write it here. So simply you label it, and the points that I am giving, that you will write in the next page. Okay. Significance. What is the major significance of glycocalyx? The first important significance that we are discussing is colonization. Colonization. What is colonization? Colonization means here A group of bacteria, normally they are unicellular, and in order to increase their resistance, their power, what they will do, children? They will come together. They will form an enclosed population. They colonize. They form a mass or a group to be stronger, and for this colonization, they will use the glycocalyx. Okay, so in order to invade the host body, in order to create an infection, this is one of the method that the bacteria is using. Colonization will happen with the help of the attachment of glycocalyx of all the bacteria, thus forming a mass of bacteria, and this mass of bacteria in biology is known as biofilm. So through colonization, what will they form? Biofilms. Or this can also be called as plaque. Colonization will result in mass of group of or team of cells that come together with the help of attachment of glycocalyx of each bacterial cell, thus forming biofilms or plaques. They become hard and rigid, almost impossible to kill them, right? So when they form biofilms or plaques, we cannot kill them easily. For example, the bacterial plaque on tooth. It is caused by Streptococcus mutans. Okay. So bacterial plaque on tooth, it is caused by, example I have given, the bacteria Streptococcus mutans. This is one of the examples. So the first important significance, they help in colonization. And what is the second important point here? What is the second important point here, children? They help in attachment. Yes, the bacteria invade the host body or any organism, or you can say some plant surface. It want to attach to its surface. First, the attachment is important, then only it can invade the host, right? So attachment will happen with the glycocalyx. So it helps in attachment to the structures like hair, nails, or you can say hair, nails, roots of the plant, anything like that. Okay, so it mainly helps in the attachment. First is colonization, the second is the attachment. The third important function is it helps in escaping phagocytosis. What is phagocytosis? Phagocytosis, it is nothing but a kind of immune reaction. Let us say for human body, if there is something called a foreign particle or a foreign object, it is entering inside the human body, what will happen? Will it be calm? What it will do? The human body will immediately create a reaction. It will show some response to invade, capture and kill that particular foreign particle. It will be observing continuously because many bacteria are being inhaled, virus, everything will be entering into our body. So there must be some defense inside us, and what is that defense called as? Immune system. So our immune system, it is working like army to us. It is like a defense mechanism. So during the mechanism, what it will do, the foreign object, it will capture and it will engulf inside and it will digest it and it will throw out of the body. That process is known as phagocytosis. Okay. So here, how this glycocalyx is helping the bacteria to escape this phagocytosis? Actually, the bacterium cell is present inside and this outer layer is a protective layer. So what it is doing, children? The bacterium, it is being hidden inside this mask. So it is protecting the bacteria so that it is not exposed. Unless it is exposed, we can create a response. So without the exposing of the cell surface, we cannot create any response and we cannot show any kind of immunity against it. So in that way, what it will do, it will hide the bacterium inside by having an extra layer. Thus, it will escape the phagocytosis. Mainly we know WBC cells, they are participating in this phagocytosis and immune reactions. So that we will see later. So all these three important points that you have to note it down. Glycocalyx, what is the significance of glycocalyx? One is colonization, second is attachment, and third is they will help in the escaping of the bacterium from the immune system or phagocytosis, anything you can say. Okay, so this is regarding the first important component. And coming to the second one, cell wall. The second component is the cell wall. So in bacteria, without any exceptions, or we can say one or two exceptions, with one or two exceptions, each and every bacteria, they have this cell wall. Not like Glycocalyx children, cell wall it is present in each and every bacteria with few exceptions. And the cell wall, mainly we will discuss with the firstly we will discuss with the chemical composition, the composition of cell wall. Composition. The chemical composition here, it is made up of peptidoglycan. What I have told you, peptido means protein. Glycan means sugar. So the cell wall of bacteria, it is made up of peptidoglycan, protein and sugar. The other name for peptidoglycan, this is very important children, regarding NEET you have questions, some questions that I will discuss. Peptidoglycan is also called as murine or mucopeptide. Murine or mucopeptide. Okay, peptidoglycan, it is also known as murine or mucopeptide. Now we will see the structure of peptidoglycan. Actually we don't need any structures, the chemical structures, it is not at your level, but still we have few questions that are being asked, it is linked to that, so I'm drawing the structure. Be aware of this structure because we have a few questions which were asked in NEET children. Let us draw. So in this way many layers are present. We make this diagram and we will draw the peptides. So in this way again, if you observe, we have like this something, and we have again NAM and NAG units here. It is not required. Just to draw this, okay. Here, peptidoglycan, it is made up of monomer sugars, and these are known as NAG and NAM. What is NAG? NAG means N-acetyl glucosamine. NAM means N-acetyl uramic acid. Okay, so we have two units which are again bonded together along with the protein chains. So these are the proteins. Okay, this is the peptide bond. Proteins have peptide bonds. Okay, this is protein chain. And in this, if you see the monomer units, NAG and NAM are present. NAG means N-acetylglucosamine and NAM means N-acetylmuramic acid. They are again in turn bonded with the peptide bonds, that is, protein chains, and this is the structure of the peptidoglycan. It is not required for you to understand what is NAG, what is NAM, but you understand that there are monomers which are made up of proteins and sugar. This is the composition of the cell wall. Now, why we are discussing regarding this, that I will tell you now. Mainly, in order to kill the bacteria, or you can say in order to control the impact of the pathogenicity, we use certain chemicals or medicines called antibiotics. We all know, we use in our daily life, we use antibiotics. What are these antibiotics, children? Anti means against, biotics means life. So the chemicals which are against life are known as antibiotics. Antibiotics are mainly playing role in controlling the effect of the bacteria or the disease caused by the bacteria. So there is one important antibiotic which is known as penicillin. One important antibiotic that is known as penicillin. And this penicillin, it destroys, or what you can say, it will mainly attack or show impact on these bonds. What it will do? It will be mainly attacking which region? The cross link between the NAM and NAG units. So this cross linking, it will be mainly attacked, showing impact on the NAM and NAG cross linking. And hence we are studying this structure. In order to show the impact of the penicillin on the cell wall, I am showing you the structure of the cell wall. And there is another natural antimicrobial enzyme which is known as lysozyme. This is antimicrobial enzyme which is present in the bodily secretions in human body, like saliva, we have tears and milk. So all these body secretions, we have the enzyme called lysozyme. It is naturally antimicrobial in nature. So it have the ability to kill the bacteria. So how it will kill the bacteria? Again, it is destroying the cell wall. Penicillin also attack the bacteria by destroying the cell wall, but where it is destroying, it is it is cleaving the cross links between NAM and NAG units. And the lysozyme, it will be cleaving the protein chains. Okay, so this is the major difference. This is important. In NEET we got this question, children, so you have to keep a star mark over here. Penicillin, it will be showing impact on the cross linking of NAM and NAG units, and the lysozyme, it is showing impact on the protein structure. Okay, so protein bonds present between the NAG to NAG units. Okay, so that's all about today, and we will see the remaining part of the cell wall in the next topic. Thank you.